Last year's ASH meeting saw what was described as jaw-dropping phase 1 results with bb2121. Now that is a second-generation CAR T-cell therapy, and it's being used in patients with relapsed refractory multiple myeloma. So that was ASH. Now we're at ASCO. Let's see where we are. To do that, I'm with Noopur Raje, who is an MD and professor of medicine at Harvard Medical School, as well as director of the multiple myeloma program at Massachusetts General Hospital. Now, to set the stage, remind us of what we know so far about bb2121, like the ASH results.
Sure. So, bb2121, as you just pointed out, Rick, is a second-generation CAR T-cell. It is a CAR which is directed against human BCMA, and what we used there to generate this CAR is a lentivirus. I think the important piece here, most CARs which are being used in the clinic right now are second-generation CARs, and what that really means is, in addition to the co-stimulatory domain of CD3 zeta, you have an additional co-stimulatory domain, and depending on the different CARs you have around, people have used different co-stimulatory domains. Some have used CD28. This one specifically is 41BB, and we really do think that this 41BB co-stimulatory domain is why we've seen the kind of T-cell expansion that we've seen with this product, and also the lack of toxicity or the decreased toxicity with this approach in myeloma.
You were a very popular person at ASH this last year. What were you presenting?
At this past ASH, we were on the verge of getting an approval for a bone-targeted agent, which we've been waiting for a long time. So we'd just gotten denosumab approved. We'd had a few other studies which we'd presented as well, where we've used RVD light. So a lot of other therapies, but mostly for the bone-targeted agent. And at this year's ASCO, as with every meeting, we seem to have new stuff coming along, which is a good problem to have.
Absolutely. And in the ASH results, they showed, for this study, they showed an overall response rate of like 94%.
Right. So, I think what this data is just an extension of what we'd presented at ASH already. What we have now is completed the phase 1 as well as the expansion. So this includes 43 patients. At ASH, we'd presented, I believe, 22 patients. So a lot more experience with the CAR T-cell program. What we have seen in patients who have gotten kind of therapeutic doses of the CAR T-cell product is close to above a 90% response rate. There is a small subset of patients who've gotten 150 million CARs, and that was in the recent past post-ASH, and we still don't have the response data. That response data, I think, is still dynamic, and in that subset, we've shown a lower response rate of 50%. I think it'll catch up. So in general, I think what we can still say at this ASCO is we're seeing very high response rates of about in excess of 90%. I think there's a few other takeaways which have been presented at this year's ASCO. One question has always been, do you need a certain level of BCMA expression to make the CARs actually work? So do you need real target expression to get that target engagement? And what we have done here is, in the expansion cohort, we'd included patients who were BCMA low expressors as well, and we've seen the same kind of data. Whether you're a low expressor or a high expressor, your response rate is in excess of 90% in this patient population. The other thing, I think, which we weren't 100% sure of was, when you look at all the toxicity profile at last year's ASH, we did present one patient with neurotoxicity, and we went into great detail about that neurotoxicity, and that made us a little more cautious about the program. Having said that, now looking at all 43 patients, we see a very nice toxicity signal in that very little toxicity. So if you look at CRS, for example, we're seeing overall a little over 60%, and the majority of this is grade 1 and 2, so not really high-grade CRS, and that is depicted by the fact that very few patients actually got the drug tocilizumab, and we use very little steroid with this. The other thing again was, we were concerned about the neurotoxicity, but when we look at all comers, neurotoxicity rate is about 30%. again grade 1 and 2, with the exception of that one patient who had grade 4, which completely resolved, and that patient is doing quite well.
So you added, what, about 20 patients, I think, to what you're presenting here?
Correct.
And you had a CR, complete response, back in ASH, I think, of 56%. What is it here?
The CR rate is still evolving, and it's close to around the same number. It's not going to change that because of the bigger numbers. I think what we are presenting for the first time now is the duration of response, which to date we hadn't looked at, but given that we have a longer follow-up of close to a year, what we have in all patients, all comers, is a progression-free survival rate of about 11.8 months. And we also, if you look at patients who were MRD negative in the bone marrow and just take those patients, what we have is a progression-free survival of 17.8 months. And both of these numbers in terms of PFS is actually, in my mind, unprecedented, because these were really very refractory patients. The number of lines of treatment these patients have had on a median is about 7 and 8 in both the escalation phase as well as the expansion cohort phase. To see this kind of a durable response was actually quite, to me, looked really quite nice.
I mean, everything's encouraging, but still, this is early data. What kind of time frame are we looking at for expanding the study?
So the expanding of the study is happening as we speak. Given that we have this kind of response data and this kind of durability, we are already doing the phase 2 trial. It's called the KARMA trial. It's accruing in North America as well as in Europe. And the plan there is to have close to 80 or 90 patients. It's there on clinicaltrials.gov. And that hopefully is going to be how this technology will get approval in an accelerated fashion, if all goes well. Along with that, we have other studies planned, because remember, this study which we presented at this year's ASCO was almost an end-stage myeloma, having received every treatment. But when you start seeing success like this, and I think the biggest concerns we had going into this kind of an approach is, are patients with myeloma going to be able to tolerate a technology such as this? I think we've demonstrated that that is possible, and then the next step would be to try and get it earlier on in earlier lines of treatment. So those studies are being planned also. A phase 3 planning towards the end of the year is something one can look forward to, and also focusing on a high-risk patient population early as opposed to late is something which we're very interested in trying to pursue.
So you're optimistic?
I absolutely. We've never seen this kind of data before in this space. So for that reason, I'm optimistic. I think one of the things which we always have to deal with and kind of temper is expectation. And I think a lot of the expectation was CAR T-cell is going to cure everybody with every heme malignancy. And I do think it's important for us to take a step back and appreciate how different all the different heme malignancies are. If you put that into perspective and kind of compare this even with the lymphoma data, the myeloma data in this setting looks extremely good. And, I think it's to consider curing a disease, unless you bring it at an earlier phase, you're not going to be able to do it.